Hello, everybody. So it's 1 o'clock. I think it's probably time we kick off. Rob, are you around? You've got your mic and video switched off, but I don't know if you're here or not. Yeah, there you
On
are.
here.
Marvelous. So thanks everybody for coming. I can see the participants are rising rapidly. We're currently at 34 and hopefully more will join. Rob is going to give a brief introduction to the day and where paediatric oncology sits in the broader scheme of the Manchester Cancer Research Centre. But I thought I would just do some housekeeping things. So there will be quite a lot of people on this call. I'm sure we're all very used to Zoom calls by now, but if I could ask everybody to mute yourselves if you're not speaking, and probably turn your videos off as well, just to save on bandwidth in case that becomes a problem. I think everybody has their name up there, but if you don't have your proper name up, could you possibly change your name so we can see who you are? So the course of this afternoon is Rob's going to give an introduction, then we're going to have a few talks by people in Manchester who are really leading areas of research which are Manchester strengths that are relevant for childhood cancer. We'll then have a small break and then we'll go on to breakout rooms, and you get to choose which breakout room you go into. And really the aim of those breakout rooms is to start to discuss specific projects where we think Manchester could take a leadership role, but where we currently don't have a lot of strengths. And then we'll come back to have a wrap-up session to think about what can we work on now, what do we need to start working for the future. So Rob, if I could get you to just give, as I said, an introduction to where paediatric oncology currently sits in the broader Manchester picture. Thanks very much.
Yeah, absolutely. I hopefully everybody can see this in just a moment. Sorry. There we go, getting there. So it's just one slide actually. And I just wanted to, first of all, thank everyone who has really driven today's workshop, and thanks to Martin, JP Kilday, and others. So I just wanted to point out again, we're in a unique position in Manchester, having the Manchester Cancer Research Centre as really a partnership between a world-leading international recognized university of Manchester, the UK's largest children's hospital, which is of course the Royal Children's in Manchester, and then also willing partners such as Christie, depending on the project, such as Proton. And where we've really been excited, I think, in the last year is to have conversations with Cancer Research UK and particularly Friends of Rosie, and John is on the workshop today, really driving to say how can we kick into high gear pediatric oncology research within Manchester, given the MCRC's piece of £25 million of infrastructure across the RadNet radiotherapy program, early detection with ACE, and a clinical academic training program that has both an MD PhD as well as a CRTF program. These are all opportunities for which we can really start to drive some of our best basic and discovery science into the clinical realm. And so it's partners like Friends of Rosie, it's the hospital domains and the clinical trials that are driven within them, and the university that does kind of have the input into the MCRC. And of course, we'll be very interested in terms of the Greater Manchester goals for children, as well as NHS goals for pediatric oncology, to be a part of those. I think this is a very important component for our MASC and Health Innovation Manchester to point out those projects that might come from invention and entrepreneurial pieces around our research, and we have great partners in that particular realm. And then really some exciting grants that recently have been given, 3 from Friends of Rosie, which we'll hear about today, and then also the recent Stand Up to Cancer CRUK grant, one of only 3 given internationally. So the MCRC sees pediatric oncology as one of the pillars of our programs going forward. We do have a CRUK center renewal coming up in June of 2021, and I certainly want to see pediatric oncology for the first time as part of that renewal. I'm a very strong zealot for pediatric oncology research, myself coming from Toronto and having a number of collaborations with colleagues who are at the Hospital for Sick Children. And the MCRC also will be supporting pediatric oncology with a seminar program starting in 2021, in which the very best in the world will be giving virtual and soon, hopefully in person, seminars, and we'll be able again to show our wares, show our research, and drive collaborations as an international collaborator of choice. So today's really talking about USPs, really fine-tuning the granularity about what we do best and what we can offer as a collaborator of choice. And Martin, I'm really excited, for the next couple of hours to see what comes through our workshops and our kind of action plan at the very end. So I'll hand back to you and I'll stop sharing. Thank you.
Thanks very much, Rob. So we'll just move on to the first speaker. If I could just remind everybody, because we've had a few more people join, if you can keep yourself on mute and turn your video off just to save on bandwidth while we're all on the talk together. So I'd like to introduce Kevin Cooper and Mike Haley, who are going to talk about unlocking the secrets of the tumor microenvironment. Thanks very much.
So let me just share my screen. Can everyone see this okay? Yeah?
Yeah, perfect.
Okay, so the title for this talk, Unlocking the Secrets of the Tumor Microenvironment. It's a little bit fanciful, but I think what we've been trying to really set up in the last year can provide new insights in terms of the architecture and the cellular composition of the tumor microenvironment. So I'm going to give a little introduction, and then Mike, who's really been driving all this work in this area, is going to go through and introduce the technology that's allowing this project to go forward and some of the results that we have been getting. And the results that we've been getting are with the brain tumor group at Salford, who we've been working closely with in the last year or so. So this is the Hyperion imaging mass cytometer that we're going to be discussing about in this talk. So this is the tumor microenvironment. This is an example of glioma, but really it can be any solid tumor that you might be interested in. We are immunologists, so we're really showing it from an immunology perspective. And from an immunology perspective, it is one of the most complex structures that you'll find in the body. Pretty much any cell type of interest, immunology cell type of interest, can be found in the tumor microenvironment. And a lot of these cell populations can really have different functions and different phenotypes. In the era of immunotherapy for cancer, really trying to understand what immune cells do in this tumor microenvironment is incredibly important, because if you can identify immune cell populations that drive progression and drive conversion, then you can try and target them and prevent that. So the immune understanding of the tumor microenvironment is incredibly important. But if you're interested in the stromal compartment, the neoplastic cells, then this holds true as well. This is an incredibly complex environment that we have to try and understand. The problem is the way that we try and understand this complexity. So there are dozens, maybe hundreds of different cell types found in the tumor microenvironment. And we try and study them traditionally this way. So we do high dimensional analyses where we take the brain tumors from experimental murine models or directly from patients. We get the tumor tissue, and then what we do is we process that into single cells. And what people will then do is high dimensional flow cytometry or CyTOF. And you get this sort of result here, which indicates all the different immune cells present and their phenotype. So it gives you an understanding of the composition of the immune cells or other cells present in the tumor microenvironment. Similarly, you can do bulk RNA sequencing, or you can do single cell RNA sequencing, and that will again tell you the different cell populations that may reside within that tumor microenvironment. The problem is that this type of approach really doesn't give you any information on the architecture or the topology of the tumor microenvironment. It tells you the cells that are there, but it doesn't tell you really where they are, and it doesn't tell you how they interact together. And sorry, I just heard some talking in the background. And this is a fundamental problem. Not understanding where the cells are and how they interact means that we don't really understand what all these different cell populations do. So we have to essentially take the tissue And instead of processing the tissue into single cells, what we really should be doing is stopping here and studying this tumor. So actually studying the tissue directly. Historically, that hasn't been possible because to study the tissue directly means you had to do immunofluorescence staining or something equivalent, which means you could only do maybe 3 channels at a particular time. New technology, such as the Hyperion imaging mass cytometer, now allows us to do much, much more. We can do 10-fold more antibodies staining on these tissues. So we can do high-dimensional spatial analyses directly into the tumor. And what this means is that in the last year or two, other groups have started to do this high-dimensional spatial analyses of tumor microenvironments. And what they're finding is that it's often not actually just the number of immune cells that are present in the tumor, which is important in defining progression or the clinical trajectory of the disease. It really is where these cells are and what they're interacting with. So for example, down here in this example out this year, it's whether immune cells compartmentalize in particular cell neighborhoods, so clusters of cells. And it's if these neighborhoods are present with the clusters of these cells that really dictates the clinical trajectory and whether these patients are at high risk or low risk of progression and death. Similarly, here, and this was a year or so ago, it's if the immune cells are mixed throughout the tumor, then the prognosis for that patient is very, very bad. If the immune cells are compartmentalized in particular discrete regions, then the actually outcome or prognosis is very, very good. So the premise is that if you take your tumor and you mash it up into single cells and just quantify the cells, you miss all this spatial insight. And often it's the spatial insight that's really defining what the clinical trajectory is and what these patient subgroups are in cancer. And with that, I'm going to stop and I'm going to let Mike take over because, again, as I indicate, he's the one who's really been driving this work in Manchester. We got the Hyperion about a year ago from a PPCRC grant. And from there, Mike has done pretty much all the optimization and driving work. So he can describe this a lot better than I can.
Thanks, Kevin. And hopefully now you can see going to my screen sharing. So I'm going to talk a bit more about the technology and what we've actually been using. And a bit sort of the practical, how do we practically use this technology? So in terms of like how it actually works, it's very, very simple to think of as immunohistochemistry, but with 35+ antibodies. So all the way you sort of go around designing the experiments or considerations of antibodies and antigens are all really similar. So we can use sort of existing knowledge there to sort of springboard these Hyperion type projects. So you take your tissue interest, so this can be a frozen or paraffin embedded tissue. You stain it with 35 upwards individual antibodies, each of which are individually conjugated to different metals at different sizes. We then, areas of tissue of interest get ablated with a laser. The plume of tissue, so this is like a helium gas, will carry this tiny bit of tissue that's been ablated, pass it through a plasma source, and then you can identify the individual metal ions that will conjugate to your antibodies. This is the technical aspects we don't really need to worry about too much. The interesting part is that we can reconstruct all these 35 different metals that we've got conjugated to antibodies into 35+ channels. So we can essentially do 35 antibodies in parallel. And from that, we can then do some really quite interesting downstream analysis to get single cell data and use all the type of analysis we normally use on single cell data. Apart from, we also have that spatial information that Kevin was talking about, the real to understand the structure of the tissue microenvironment. So currently, there's only 35 metal tags currently used, but in theory, this is the upwards of 135. So we're limited only really by the manufacturers are limited by the availability of the pure metal ions they can get, and also whether we want to make sure the metals we use aren't already in other biological systems. So the ones we use, these metals aren't present in biological systems, and also that means we don't have to worry about autofluorescence, or there's essentially no background other than due to the antibody staining. And the channels are all very discrete, so you get very, very, very little bleed through from one channel to another, which is something which is a consideration if you're using fluorescence approaches, especially if anybody has ever tried to multiplex of upwards of 7+ colours, it's quite tricky to do deconvolution. So we've been using this, the Hyperion technology, to try and understand sort of several different types of brain tumour. So these are adult cases, but as Kevin said, the principles still apply to any type of tumour really. So I've been using rather to look at these low-grade astrocytomas and try to understand how and why these low-grade astrocytomas, which are sort of like relatively symptom-free for several years, and then progress into these highly aggressive tumours, which are rapidly fatal. And we don't really know how or why or even when these tumours may progress. So these patients are unfortunately left with these sort of ticking time bombs of the progression that can, obviously quite troubling for them. So we want to understand the mechanisms how this transformation occurs. So first of all, we started off with a glioblastoma case to try and sort of get the ball rolling, and we got some really nice early results. So we just took paraffin-embedded sections from a case of glioblastoma, and we could really quite clearly identify all the different cellular components that we'd expect to see in these cases. And the actual picture you get looked very similar to what you'd see for normal IF. So here I'm only showing 6 of the colours we stand for. Clearly, putting 35 different colours on one image, it's not possible, there aren't enough colours. But you can sort of see visually that it's what I quite expect. However, this isn't sort of the end of the story, really. This isn't the limit of the technology isn't just getting pretty pictures, it's then getting that downstream analysis. So we wanted to then actually find a case of astrocytoma to look at these mechanisms of progression. And we wanted to get a case where we had sort of low-grade regions, high-grade regions, also healthy grade regions in the same patient, to try and compare these regions using the Hyperion. So we got all these cases on the same slide, so we probably stay at the same time. And then we got a pathologist to identify the regions on a H&E stain section. And then we use a serial cut section to do our antibody staining on. So we go in with our antibody panel, we stain the section, put it into Hyperion with the region of interest. And the output of that is this high dimensional imaging. So we get sort of 35 different channels imaged. So this is the type of data we see. So it's from the high grade region, the low and healthy control region from the same patient. So you can see we can really kind of stain for anything you can normally target by normal image chemistry. So hopefully it's sometimes quite, and this is part of the problem when you have so many colours, it's quite difficult to see exactly what's going on. But hopefully you can see that we can really get the different components of the tissue microenvironment. So there's a huge number of myeloid cells that infiltrate in these cases in both the low and the high grade. We see T cells, and we can also look at signaling molecules, phospho antibodies. So we really can look at several different targets, get a lot of information about the cellular composition. But how do they go from all these 35 different channels to get some usable data at the end? How do we get that single cell data? So, besides the antibody channels, we also have a nuclear stain, this iridium metal. So this can be used to identify the cells, individual cells in the tissue sections. And then we segment the image into individual cell masks. So we do this using cell profiler, but this has been sort of superseded by machine learning type approaches that are a bit perhaps more accurate. And I'm now using to identify these single cells. This can then be applied, this gives you a mask you can apply to all your channels, which essentially gives you single cell expression of all your markers. Though critically, we've also got, as I said, the position of these cells within the tissue. So we've got individual cell expression of all the markers and whereabouts all those cells are in the tissue. So I've used this sort of more downstream approaches to try and to start comparing between some low and high grade regions within this particular case. So it's worth pointing out that we really are just scratching the surface of these type of techniques, and there's a huge amount that I'm still to learn yet to really try and get the most out of these tissues and data sets. So this is quite a dense slide, so let's quickly talk through it. So I've done analysis of a high, low, and a healthy Hyperion data set from this patient. On the left here, you can see cell clusters of the different cell types identified. On the right here, you can see the full antibody panel. So these are all populations of cells that were identified using a clustering algorithm called phenograph. So this is sort of an unsupervised approach. We didn't tell it what populations to find. These are sort of populations that sort of jump out of the data set due to the sort of discrete nature of staining. And so we can see populations you expect to see here, such as vascular cells, which are quite easy to identify, lymphocytes, some different neoplastic populations, and myeloid cells as well.
Specifying the hospital, but usually it'd be either.
So the myeloid cells we're particularly interested in. But it's quite clear from this analysis that when we sort of compare the high versus low grade regions, there's a drastically different cellular composition. And this isn't entirely surprising, but it shows we can come to that conclusion in like an unsupervised fashion. So we really want to think about how can we use the spatial information as well. So we did sort of like a more specific analysis to look at the myeloid cells specifically, because I think these are really important in this sort of transformation of these low grade to high grade cases. So you can see these sort of like these 3 markers for myeloid cells, and just by expressing these three together, you can see there's a huge variety in the myeloid cell composition here. It's not like they're all expressing the same. There's a huge amount of variety there. But then if we we can actually map back the populations we've identified using the phenograph and using these unsupervised approaches, we can map them back to whereabouts they are in the tissue. So this is quite a preliminary sort of finding here, but it sort of illustrates the potential observations you can make. That the myeloid cells we think are potentially infiltrating monocytes are actually localized around the blood vessels, which is what one might expect for infiltrating cells. And potentially also see some particular niches where these tumor-associated myeloid cells might be localizing. So it's quite clear that sort of these myeloid cell populations have different locations of the tumor, and that's what the Hyperion can tell us. So this is another type of intracranial tumor we've also been working on with in collaboration with Salford, the mentioned school-based group there. So these fibrinomas are benign intracranial tumours, but they're very interesting from a sort of immunological point of view. Because what the school-based unit have found is that inflammation is really important for promoting the growth of these tumours. So these tumours can be actually static for quite a long time. So these patients, they can have static or growing tumours. And what they found is that the growing tumours have a huge infiltration or proliferation potentially of IBA1 positive myeloid cells within the tumours, suggesting that inflammation is really important in these tumours for driving their growth. So we want to use the Hyperion to try and understand this finding a bit better. Can we get a bit more of an insight into these IBA1 positive cells? So we already know they're clearly more abundant in the growing cases, but are they also a different phenotype, which suggests having a different function? So we could use the Hyperion to identify all the different cell populations we expect to see, and it works quite nicely. Again, this is paraffin-fed tissue with regions of interest identified by a neuropathologist. And we can see immune cells that we expect and other markers of interest. So we can then use a similar type of pipeline you use for the astrocytoma case to try and compare the static versus growing tumours, to try and look perhaps more specifically at whether the IBA1 positive cells have got a different phenotype. And it looks like that's the case. So when we compare the growing versus static case, the IBA1 positive cells and the growing versus static cases, we can see that the populations are quite distinct. So the IBA1 positive cells in the growing cases seem to be a lot more inflammatory compared to the IBA1, compared to the resting or static tumors, which have an IBA1 positive phenotype. It's a lot more resting, less inflammatory. So this sort of like suggests that the phenotype between these static tumors, the phenotype of the myeloid cells in these different tumors is completely different. And this is just to show we can sort of correlate spatially these different regions, so we can sort of map back all these populations whereabouts they are in the tumour. But clearly, this sort of the type of analysis, these spatial analyses I've been talking about then, really are just scratching the surface, the power of what we could potentially do. So we want to, you can really build up this spatial information. So, you know, this spatial information is the powerful part. So we can do some more in-depth analyses in the future. So I'll look at neighborhood analysis to try and understand whether certain cell populations associate with each other, and then build that up into sort of populations of networks of different cells that interact. And whether cells are differently spatially distributed without tissue, and whether we can see a sort of trajectory of certain cells having different stages of differentiation in the tumour. And these type of analysis are exactly what Kevin was referring to earlier. Sort of using these sort of in-depth spatial analyses to identify different tumour categorizations, so these sort of mixed versus compartmentalized tumour in this breast cancer cases that Kevin was referring to earlier. And when other groups have done these type of analysis, it becomes really clear that you can see different types and structures of tumor microenvironment that are really critical for defining survival and recovery. So I'd like to thank Kevin and supervisor that's won the funding to get the Hyperion. It's been and the Manchester Centre for Clinical Neuroscience and everyone else that's on for the support of this work, and the Force facility, Gareth Howe specifically. Thank you very much.
Thank you, Mike. That was some really lovely data that you've shown there. We do have a few minutes, actually, if anybody has any questions. Can I ask, if you have questions, can you put them in the chat, and we'll just keep an eye on them and pick them up. Can I just kick off? So, I mean, I asked you to talk about immune function, obviously, because, you know, immune therapy is a real growth area in oncology, and it hasn't really hit paediatric cancer very much. But about the technology, particularly Hyperion, how limited are we by the antibodies? Are the antibodies really designed around immune cells, or can we stick the metal ions on any antibodies we want?
Absolutely, anything we want. So really, this sort of you can attach anything you could detect with an antibody, anything you can really attach a metal to. So people have really used this for you know, attaching to antibodies. But groups have also done things like looked at where platinum-based chemotherapies end up in the tumour and things like that. So it's not limited. It really is the sky's the limit really here. Although our panels and our focus have been on sort of the immune targets, really it's anything you can target with antibody really, or any other way you can target a metal to certain parts of the tissue.
Okay. And do we make the antibodies here, or do we buy them from another country?
So you can conjugate. So Fluidigm have their own panel, own readily validated panel antibodies. So they've got a range which are, in theory, pre-validated to work on most paraffin-embedded human tissue. And we found that they're pretty good. But we can also conjugate our own antibodies. So you can conjugate whatever antibody you want to whatever metal you want, and you can build up a panel that way, which is quite a big task to build the panels. But we've already sort of got a lot of antibodies that we've optimized and worked up in human tissue, which is a good starting point.
And is it limited to paraffin-embedded tissue, or can you do it, for instance, in liquid biopsies or frozen?
So you can do if the liquid biopsies, you could probably do a suspension site off, which would probably be better. But in theory, you can do it on smears and things like that. So it's only limited to, you can do it on frozen tissue. I think some people have done it on smeared cells as well. So like for liquid biopsies, they've then smeared on slides and things like that. So anything you get on a slide really is the answer to that.
Okay, brilliant. I mean, it's some really lovely work, and I'm sure it's going to be very important. I can't see any other questions coming up on the chat. I mean, we have a session in the breakout sessions on exactly this area, so hopefully you guys can stay around. Kevin, I think you've got to disappear, but you're hoping to come back.
I'll try and come back, yeah.
Great, marvellous. Okay, thanks very much, Mike. So the next talk we have is from Sam Butterworth, and this is about transcription factors. We thought this would be a good area to think about, because we know that a lot of paediatric cancers are driven by either transcription factors being overexpressed or transcription factors being bound to something else in the fusion protein. And these have always been historically virtually impossible to target. So we asked Sam Butterworth if he'd be able to give us an update on whether there are new ways to target these now and where we are up to with the transcription factors. So Sam, are you? Yep.
I am here. Yep.
Marvelous. Great.
Just stick my presentation up. Hopefully that will work.
That's it.
Marvelous. So yeah, so this is more of, I guess, of an update and something of an opinion piece really around the potential for targeting transcription factors, and particularly, I guess, mutated or transcription factors or transcription factor fusion proteins, etc., for treatment of particularly pediatric oncology. So I'm going to start off with just, I guess, a very high level overview of the mechanisms in which we, I guess, now know how to target driver mutations in oncology setting more broadly. And I guess the three levels at which most drugs target the cancers are either by directly targeting the mutated proteins themselves, looking at some kind of secondary targeting of changes in signaling or regulatory processes in those cells as a result of that mutation, or ultimately targeting the phenotypic changes in the cells themselves. And I guess inevitably what we're looking for here then is a mechanism in which these drugs are able to distinguish the cancerous cells from healthy tissue in the body in order to generate some kind of therapeutic window. So obviously the kind of classic approach to killing cancer cells would be through sort of cell cycles, cytotoxins. So really ignoring the mutational background of the cancer and focusing on the fact that cancer cells are dividing faster to distinguish them from healthy cells. And obviously that's the kind of basis for conventional chemotherapy. I guess the initial sort of examples of really targeted therapy, drugs like imatinib, are drugs which are able to directly target driver mutations, for example, ABL fusion proteins in this case. However, a drug like imatinib actually isn't particularly selective for the mutated protein itself. It targets ABL and has selectivity for cancer cells because that protein is so important to that subset of cancers that you can get a therapeutic window. Whereas some of the more recent drugs targeting driver mutations, again, looking at some kinase examples, but there are others, for example, BRAF and EGFR, these drugs are able to achieve selectivity for the tumor by being intrinsically more active inhibitors of the mutated protein than they are the wild type protein that's expressed in the rest of the cells. And this can potentially allow you to drive really much more significant therapeutic windows for these kinds of drugs. Obviously, not all cancer drugs target the mutations themselves. So if we look down at the bottom, I guess there's a whole emerging area around molecules that target somewhere between sort of signaling, regulatory changes, and phenotype. So thinking about targets which are up or down regulated secondary to mutations. So HER2 in breast cancer would be a really good example of that. Cells that can target the phenotype in terms of the tumor microenvironment, which we've just been discussing. So sort of immunotherapies, obviously strong emerging areas we've discussed. And perhaps another really very strongly emerging area around antibody drug conjugates that sits somewhere between these two, using typically conventional non-specific cytotoxins, but targeting proteins which are overexpressed on the surface of cancer cells as a result of the regulatory changes. So again, often not mutant specific, but distinguishing cancers from normal cells based on the proteins that they're making as a result of the mutations that they're carrying. And then I guess a few other more recently emerging areas, so around, for example, synthetic lethality, so mechanisms where mutations, particularly in DNA damage response, can enable or make cancer cells very sensitive to inhibition of other aspects of the DNA damage response pathway. So PARP inhibitors in BRCA mutation is a really good example of this. So again, you're not targeting the mutation directly, but you are getting potentially quite high selectivity for cancer cells bearing specific mutations. And I guess one example of very relevant to today of a recent drug approval for drugs targeting the signaling pathways around mutations is the recent approval of a MEK inhibitor for the treatment of NF1. So this is basically disrupting the signaling pathway downstream of the mutational driver in a way that seems to give you sufficient specificity to show efficacy in this setting. So this is kind of a high-level overview. Thinking about what this means then for transcription factors, and particularly transcription factors relevant to paediatric oncology settings. So this is an example of one of the, I guess, well-established fusion proteins that's a driver in subset of paediatric oncology, this PAX3 OXO1, and this is taken from a paper. But what you can see here is effectively the mutation causes the formation of a transcription factor which has a differential recognition of DNA and therefore results in dysregulation of transcription. And quite a lot is understood about the signaling pathways around this as a target. So people have looked at potential to inhibit the formation, the synthesis of the protein, its activation by kinases, its maintenance and degradation by other kinases, the mechanisms by which it interacts with DNA through epigenetic modulation, etc. And then ultimately, once this transcription factor binds to DNA, it causes altered expression of a wide range of genes, many of which are potentially targetable as drugs. So when you've got a mutated transcription factor, you can think about addressing this, as per the previous slide, at, I guess, multiple different levels. So what I've done is had to think about what are the implications of that then for targeting transcription factors. So first of all, and this is, I think, the comment that probably Martin has received multiple times, and it's really what today's talk is about, is these transcription factors themselves are hard to directly target. They often don't have what we consider as being sort of druggable pockets, so regions on the protein that it's easy to imagine how you're going to modulate the protein by binding to. This can become even more difficult if there are any inactivating mutations, although I think in the case of paediatric oncology, almost all of these are activating mutations, which does make it easier to imagine inhibiting, reactivating a protein can be more difficult. And coming back to the point I made on the previous slide, one of the challenges would come even more strong if you really needed mutant specificity in order to have a therapeutic window. So obviously the individual components, the individual transcription factors that comprise these fusion proteins themselves have often very fundamental physiological function. And if you need to leave those alone, then the challenge of disrupting the activity of just the mutated transcription factor becomes even harder. Of course, on the flip side, where these drive mutations are occurring on cells with relatively low total mutational burden, there's obviously a huge potential for therapeutic benefit if you can target those, potentially have really transformational effects for patients. When we look downstream or upstream, and this comes back to some of the data on the previous slide, the challenge here can be that often the mechanisms that regulate the behavior of these transcription factors are themselves also quite fundamental, and the targets identified on that previous slide, for example, I don't think any of those are ones you would go after confidence of being able to find a drug that had a decent therapeutic window. And they have also, because their transcription factors, often quite a broad impact on gene regulation downstream, and so trying to pull out specific components, specific targets downstream of those transcription factors and targeting those is also going to be challenging. And I think the point here is that it's going to require really, I think, a really deep understanding of which are the key aspects of the signaling pathways around these transcription factors that can be targeted before you can really come up with a hypothesis for a specific protein or family of proteins that you want to go after. So I think this is almost still a biological problem, really, so much as a drug discovery one. The good news, though, after all that negativity, is that in recent years, there have been really huge advances in the drug discovery community across the world in our ability to target proteins that have previously been considered undruggable. So when I started in AstraZeneca in the pharmaceutical industry 15 years ago, all three of these proteins were on the list of things that AstraZeneca would definitely wanted a drug against. So KRAS, SHP2, and STAT3. And all of them were considered undruggable by small molecules at the time. KRAS probably is the biggest example of a well-identified oncogene, which the pharmaceutical industry has been hunting for drugs for decades. through direct and indirect modulators, etc., etc. And it was seen as impossible until someone did it. And a group in America from Scripps published the first example of a KRAS inhibitor. And once we found one, it became easy actually to find others. And now there are multiple, both mutant, very specific, mutant-specific and pan-mutant compounds in clinical trials. So this just goes to show that the concept of undruggable is somewhat time-bound, at least in some cases. SHP2 is another really good example. So there are still no good active site inhibitors of this phosphatase, SHP2. However, Novartis ran a very clever and well-conceived high-throughput screening campaign that resulted in identification of allosteric inhibitors of this phosphatase. And again, once you know those compounds exist, now multiple companies have SHP2 inhibitors going into clinical trials. However, it's worth probably making the point at this point, these drugs are very unlikely to be active against the mutational drivers in SHP2 from some of the pediatric indications like JMML. And finally, and probably most relevant for today, STAT3, which is itself a transcription factor. It has no obvious druggable pocket, albeit it does have some phosphotyrosine recognition sites. And again, this, you know, if you'd asked people 15 years ago, this would definitely have been a protein that we wouldn't have known how to target. But now direct STAT3 inhibitors are entering clinical trials. So I think my summary from this is that great things can be achieved, but you need to have the right biological models. You know, all of this work has come from very, very complex screening cascades typically, and you need the right compound collections. There's no point having a good assay if you don't have the right compounds. And this is, of course, a challenge for academic groups. I'm going to touch on two more things briefly relating to this. So these KRAS, the SHP2 and STAT3 inhibitors have been published over the last few years. But there's a technology that has been, I guess, really gaining a lot of momentum in the drug discovery community, which has since been applied to all of these targets, which are these types of molecules known as PROTACs that drive the targeted degradation of proteins. So I know some of you on the call are aware of this mechanism, but I'm just going to touch on it briefly for anyone who isn't. So a conventional inhibitor of or modulator of a protein effectively binds to that protein. So if you've got the protein here in blue and your inhibitor in gray, through some kind of equilibrium that brings the protein into in an active state typically, or potentially an active state if you're looking at an agonist. So this can be seen as sort of occupancy-driven pharmacology. You need enough drug present to switch on or off enough of the protein to see an effect. So some work from the Cruz group in Yale identified now 20 years ago, but really in recent years have there been compounds which are seen as suitable for use as drugs, a new approach that they termed PROTACs. And these are heterobifunctional ligands. So now these molecules bind to the target protein that you're interested in, but they have a second domain that can recruit an E3 ligase. And that ligase complex results in ubiquitination of your target protein. So effectively what you're doing is you're hijacking the natural protein turnover mechanism of the cells, and you're using a small molecule to force the ubiquitination of a protein that this ligase complex wouldn't usually target. And that results then in proteasomal degradation of the protein without actually breaking down the ligand itself, which is able to then go around and repeat this process. And this then targeted degradation can allow for the effectively removal of cells, almost like using siRNA of a protein, but now using a small molecule. So I mentioned this approach has been applied to STAT3. So this is a publication from last year. This is still preclinical work, but just gives you an idea of how this mechanism can work. So here's a STAT3 inhibitor. It's not a very small molecule, but nonetheless, it's quite an interesting compound. And what this group have done is, in order to convert this into a PROTAC, they've added a linker and a ligase binder, one we've also used in our group, to recruit cereblon. So this is actually a thalidomide analog, binds to the same biological target as thalidomide exhibits its activity through. But this causes then the formation of a new, I guess, complex between STAT3 and cereblon complex. And this results, as you can see here, in effectively complete degradation of STAT3. And this is data from an in vivo mouse study bearing a tumor looking at a relatively low dose of this drug, and within sort of 6 to 24 hours, you can see the protein is effectively completely ablated from the tumor. One of the beautiful things you can do with PROTACs is you can use whole cell proteomics to understand what has happened in these cells. And you can see here effectively this molecule is removing STAT3 from these cells almost completely without having any really significant effect at all on the abundance of other proteins, a tiny bit of degradation of another STAT isoform. And in this case, although this is a fairly undrug-like molecule in common with all PROTACs, they've gone through to really show quite spectacular in vivo efficacy. So this shows that you can target transcription factors with these new kind of methodologies that I think just, we didn't really know about, and we certainly didn't trust until recent years. The PROTAC approach can be very powerful. Unfortunately, it still requires a ligand to start with. So the need to identify a compound that binds to the targets you're interested in doesn't go away. And these molecules are not particularly drug-like, and the development probably isn't going to be as easy as it might be with another type of small molecule drug. However, it's worth noting that there are now PROTAC compounds that are showing effects in clinical trials in cancer patients, particularly looking at estrogen and androgen degradation. So this is a path that is definitely viable. And the reason I mentioned it is one of the big advantages it has relative to, I guess, conventional occupancy-driven drugs is that in the case of the transcription factors, you don't necessarily need to find a functionally active ligand. So your molecule that's going to cause the complex to form with the ligase doesn't itself need to have functional activity in order to potentially allow you to form a PROTAC from it. And that opens up perhaps some alternative screening approaches, for example, using a technology called DNA-encoded libraries. And we have recently received a grant myself with Newcastle and Dortmund from the Wellcome Trust to set up an academic DNA-encoded library screening resource that we could think about applying to targets such as transcription factors with the right biological collaborators. Although that grant won't be really running for a couple of years because we've got to do a lot of setup work. But these DNA-encoded libraries are already available out there in the public domain as well, and can be accessed through collaboration with some of the providers that do provide preferential access for academic groups, albeit not free. One other thing I'm going to touch on that, and this is just very much an example that's something that the pharma companies, particularly AstraZeneca, we were very keen on, work from Ali Tabasoli at Southampton, who came out of the Benkovic group from his postdoc. And here they're using bacteria to randomly make assortments of huge libraries of cyclic peptides based on natural amino acids. And then they set up a reverse 2 hybrid system in bacteria that effectively suppresses the expression of survival genes in those bacteria in the presence of a complex which they're engineering into this cell. And then they get the bacteria to co-express these two, I guess, components. And what happens is effectively that the bacteria only become viable if they express a cyclic peptide, which results in the breakup of these interactions. Most of the work is done on protein-protein interactions, but you could imagine trying to configure this to look at other types of interactions as well. And the consequence of that is then they're able to effectively select out bacteria that express biologically active cyclic peptides, and then use sequencing to pull those peptides out and develop them from the bacteria. And this has proved to be a very powerful way of identifying modulators of protein-protein interactions, which have just really been completely intractable to kind of conventional drug discovery approaches. So this is just one approach, but I think alongside the DNA-encoded libraries and the PROTACs, etc., it doesn't make transcription factors easily druggable. That would be a lie. But it certainly means we have the power to do things now that we couldn't do 10 or 20 years ago, and drug targets that we were seen as being undruggable are starting to come closer to being in scope. In addition to kind of finding small molecule modulators, obviously there's the potential to go after RNAi approaches. There are some publications showing the potential for mutant-specific inhibition of protein synthesis, although I think we're going to rely a little bit on luck as well as judgment to find those. However, it's worth pointing out that targeting of solid tumors does remain difficult for kind of oligonucleotide-based approaches, and again, a lot of people are working on that problem with nanomedicines, etc., but it's still difficult to get that to go away. One of the collaborations we have in pharmacy with AstraZeneca, NowCAD, looking a lot at RNAi delivery to tumours as a potential path forward, but it's still early stage work. Thinking about signaling and regulatory changes in relation to the immune system, is it possible that the changes we're seeing driven by these transcription factors result in changes of cell surface protein expression that could be targeted by immunotherapies or neoantigens that might make these tumours more susceptible to, specific CAR T cells, etc. Remains to be seen. I think that's a biological question again that could be asked. Obviously, ADCs would be another approach to targeting those. And then a final thing I'd mention is obviously one of the perhaps most common approaches to these kind of problems in academia historically has been to run kind of phenotypic type screens on cells. I have my own reservations about those, but that's something we could discuss in the breakout room if people want to think about that approach. So yeah, so a few ideas from me to finish up and items to discuss. I think, that it's certainly possible now to think about developing screening approaches to identify novel chemotypes, but it's not easy, but it's certainly possible. We could think about genetic approaches, understanding biological pathways, etc., and how we could drive that understanding to identify new mechanisms, and taking that on to think about, neoantigens or surface proteins that could be targeted by ADCs. We have a collaboration with LifeArc in my group looking at ADC warheads, that things that we could do in that space probably. There's obviously the potential to do phenotypic screens, and I'd be happy to talk to people about that, even though it's, like I said, it's not, I don't think it's the best way forward. And I'm sure there are other ideas as well. This was just a bit of a brain dump from me. But I suppose one of the key points I think we should consider here is these are big problems that are going to need, I think, big solutions. And Manchester, maybe we could do some of these things by ourselves, but we're probably not the world leading group in any or all of these technologies. And I think it's going to be thinking about how we can collaborate and which aspects of these programs we would want to lead on if we felt it was the right thing for us to do.
That's great. Thanks very much, Sam. That's a really interesting and thought-provoking talk. There's one question. I think we're not going to have time for lots of questions, but a question from Bernadette. See, the example you gave of alveolar rhabdomyosarcoma, the multi-tyrosine kinase inhibitors obviously have lots of on-target effects, but lots of off-target effects as well. How can we find clean multi-targeted drugs?
Get lucky, probably is the. I mean, if you know the profile you want, you could probably do and you're talking about kinases, we could probably, someone like Richard Bryce and Leach, do quite a good computational analysis of whether there was, any reason to believe whether a profile is possible. And there is a lot of pan-kinome screening data out there in the public domain as well that you can mine to look for whether the kinds of profiles that you want are available. So it's, like I said, you're going to require a bit of luck. I mean, most of those dirty TKIs, if you want, are they weren't designed to have any particular profile. They were the discovery of their profiles and elucidation of how that drove efficacy really came after those compounds went into clinical trials, generally not before. So if they work, it's perhaps just by chance, and I think often probably just survival bias. The ones like Sutent that got through look good, and all the ones that fell away you kind of maybe overlook. Yeah.
Okay. One last question, quick yes or no. Do PROTACs cross the blood-brain barrier?
Short answer, some of them. Okay. Arvinas, the company that came out of Craig Crew's lab, are focusing almost entirely on CNS targets, following their early work in oncology. So it's possible, they can be so potent that you just don't need very much drug. I mean, you can get really spectacular efficacy with PROTACs in some cases, but they may not fall out. But we've had a compound that degrades its target protein with an EC50 of about 0.1 nanomolar in our group with the PROTAC methodology. So you might not need as much drug as you typically would. Okay. Yeah, if you wanted to make a drug that was definitely going to cross the blood-brain barrier, you probably wouldn't choose a PROTAC as a place to start.
Okay, I think we'll probably discuss that more in the breakout group. Thanks very much, Sam. So moving on, the next talk is by Fiona. Fiona, I'm not sure if you're doing this as a joint talk with Rob or doing all of it, but this should really have gone with Kevin and Mike's talk at the beginning, but we had to do a bit of schedule shuffling. So Fiona's going to talk about the clinical side of immune therapies.
Yeah, apologies to Rob. I never quite managed to get in touch with Rob, so I'm going to go it alone anyway. But hope you can hear me and see my slides okay. Is that okay, Martin?
Perfect.
Yep. So I'm going to start with a disclaimer, actually. I'm not going to talk as per the agenda on immunotherapies for childhood cancers, because I guess I treat adults. But what I am going to talk about is some of the really exciting advances that we've seen in adult immune oncology that have seen step changes in the way that we treat patients, and I think where there's potential applicability to childhood cancers, or where these are already being applied, actually. So if we see if I can progress my slides, which aren't progressing on my side. Let me just try. I'm going to try has
that's moved on. Yeah,
that's moved on. Okay, I'm not quite sure what I did there, but I'll keep trying that next time. Okay, so I think everyone's probably familiar with the concept of the cancer immunity cycle, and this shift that we see from the immune system being able to detect tumour antigen and eliminate it to a state of equilibrium, and then ultimately escape of the tumour, such that the immune system is no longer controlling the cancer. And this slide just shows the cancer immune cycle in a slightly different way, but what it does is flag up some of the areas that we can look towards to intervene and to have a positive effect when we're trying to re-establish that elimination phase of the cancer immunity cycle. I'm not going to talk about all of these today. In fact, I'm not really going to touch on vaccination at all. What we've seen particularly in childhood vaccinations around things like cervix cancer or prophylactic vaccinations being very effective, particularly where there's viral antigens to target. And also I'm not really going to touch on cytokines. These have been used in adult tumours such as renal cancer. We used historically a lot of high-dose IL-2 here and saw some great efficacy, but at the cost of really significant toxicity. What I will come back to is targeting specific tumour antigens, primarily through T-cell therapy. But I just wanted to touch first on the interference with the checkpoint regulatory pathways, primarily PD-1, PD-L1 pathway and the CTLA-4, because this is really where in adult oncology we've seen an enormous step change. Checkpoint inhibitors really are one of the pillars of treatment alongside chemotherapy, hormone therapy, small molecule targeted therapies and monoclonal antibodies. So they're used regularly as standard of care across multiple tumour types, particularly melanoma, lung cancer, renal cancer. And I'm sure most people are familiar with the type of mechanism involved. We have cytotoxic T cells can detect antigen via the TCR receptor presented by MHC on the tumour surface, and can then activate the T cell to cause killing. It's clear that signal to be fully effective requires an amplification signal, so an activation signal, such as B7-CD28, or and to fully activate. But if people were permanently in a state of T-cell activation, it would clearly cause issues with autoimmunity, and so then there's a negative downregulatory signal. In this case, CTLA-4/B7 engagement, upregulation and engagement, and PD-1, PD-L1 pathways. So the therapeutic agents that have come to the fore in the clinic have been primarily inhibitors of these negative signals, so blocking the blocker, as it were, to reactivate the positive signal from the TCR-MHC antigen interaction. So first of all, we had, first came the CTLA-4 antibodies, and then latterly PD-1, PD-L1 antibodies. And these, as I say, are commonly used in our clinics day to day. There's been a lot of investigation around other, in this case, on this side of the diagram, inhibitory receptors, mostly used in combination with the standard of care checkpoint inhibitors. And similarly, there are activating receptors and agonistic antibodies towards these pathways have also been used. But so far, other than the PD-1 CTLA-4 pathway, we haven't really seen a step change in activation. So what we see as we combine more and more of these antibodies is increasing toxicity without necessarily the step change in increasing efficacy. And of course, these antibodies are essentially non-specific. They're trying to release the brakes on a generalized anti-tumor immune response. And with that generality comes challenges in terms of identifying biomarkers. And clearly, there's many factors involved in activating or reactivating the immune system. And this diagram just highlights the multitude of different factors, from tumour cell intrinsic factors such as PD-L1 expression, the tumour microenvironment, where factors such as T-cell infiltration can play an important part. Those hot tumours where there's lots of T-cells already migrated to the right place are much more likely, if the brakes are taken off those T-cells, to induce an anti-tumour response. And then systemic factors such as gut microbiome, peripheral blood composition all clearly play a role. And there isn't any one biomarker which can predict highly which patients will respond. And the best we can do probably at the moment is looking at things like mutational tumour burden, PD-L1 expression, and that T-cell infiltration. But all of these factors actually are relatively unusual, particularly the mutational load in many childhood cancers. And I think this is a slide that many people have probably seen already. So it's taken from a paper from around 3-4 years ago now. And what it shows is different tumour types and their neoantigen or somatic mutation prevalence across the different tumour types. So higher, each dot represents a single patient, and where it's placed on the y-axis relates to the mutational load within that individual patient. And what you can see from this is that those tumour types where we tend to use checkpoint inhibitors and see a relatively good proportion of patients responding, melanoma, lung, bladder cancer, tend to be the tumours where there's a high mutational burden. And whereas at the other end of the scale, we start to see the malignancies of more common in childhood, medulloblastoma, neuroblastoma, glioblastoma. So we see fewer of those neoantigen, high mutational burden tumour types there. And so it's perhaps not unsurprising then that those general stimulators of checkpoint inhibitors tend to have been less effective in childhood cancers. But that's not to say no immunotherapies will work. And as I mentioned, these are potential where we might be able to identify more specific tumour antigens that can be targeted with immunotherapy approaches such as CAR T therapy. And I'll come back to that. Thinking now then, moving away from the checkpoint inhibitors and thinking more about other approaches such as cell therapy. Adoptive cell therapy has a number of real attractive features that makes it worthwhile considering. T cells are a living platform, essentially. Those T cells can hopefully migrate to the tumour site. They can be activated by endogenous factors to be activated against the cancer, and they can multiply and go on to kill multiple tumour cells. You can gene modify T cells ex vivo to make them specific for the tumour antigen of your choice. And you can stimulate the cells if they're ex vivo, so you can take the cells away in the laboratory, stimulate, activate them, grow them up to large numbers. And meanwhile, you can give the patient conditioning chemotherapy in an attempt to reduce some of the immune suppressive factors that are available within the tumour microenvironment. And it's this sort of information that's meant that adoptive cell therapy has become increasingly attractive over recent years. So we've seen a huge increase in the number of cell therapy trials that have been registered. this was part way through 2019. There probably has been a drop-off since then due to COVID and a lack of ability to roll out clinical trials. But you can see from this that over the recent years there's been an enormous interest, and that the approaches are largely T-cell based, although there's increasing interest in other cell types such as NK cells. But we're looking at things like CAR-T, TCR T-cell targeted, and TIL therapy. And I'll talk a little bit more about those in a minute. So yeah, actually in this slide, I'll talk about those a bit more. So the approaches to adoptive cell therapy fall into two main buckets when we're talking about T-cell therapy. Thinking about natural T cells, so those are T cells that are isolated from solid tumours. So a patient would have to undergo an operation to remove a portion of metastatic, usually metastatic disease, which may be from skin nodules, for example, in melanoma, or lung metastases. And then the TILs, the tumour infiltrating lymphocytes, can be grown out from those and expanded up in the laboratory. And then there's the bucket of genetically engineered T cells, of which CAR-T is one form, and then TCR the T cell receptor T cells, gene-modified T cells. But the principle for all of these treatments is very similar, and that's that you somehow need to collect the T cells, whether that's from a surgical specimen or for CAR-T and TCR T cells, usually done by apheresis. So peripheral T lymphocytes are taken. They may then be genetically modified, for example, with a retrovirus or a lentiviral vector, expanded, and meantime the patient's given conditioning chemotherapy to nullify or to reduce the immune suppressive microenvironment primarily, and then the cells are given back. And the most kind of the poster boy, as it were, for adoptive cell therapy has largely been CAR-T therapy, and most of the clinical trials and those standard of care products are what we call second generation CAR-Ts. So this is the receptor, the genetically modified receptor structure that most of these CAR-Ts use. So there's an antigen binding epitope, often an antibody fragment which can detect the antigen on the surface of the malignant cell, a transmembrane domain, and a CD3 zeta T cell signaling domain, such that when the antigen is recognized and bound, it induces signaling through the T cell to activate the T cell and for it to become cytotoxic and kill the tumor cell. The second generation bit is this part, the co-stimulatory molecule, which always surprises me slightly that even though normally and endogenously it would be a separate receptor, if you stick it in the middle of this receptor, it seems to work and amplify that signal so that you can get better engraftment, persistence of the cells, and better activation and killing. And over the years, many different types of tumour antigens have been identified and tested using this approach. A big stumbling block, particularly in solid tumour oncology, has been that if you try and target with CAR-T, antigens that are overexpressed on cancer compared to normal tissue, you often see cross-reactivity, even to low-level expression, such as with HER2. There's been some issues with cardiac expression and cardiac toxicity originally, although there's since been subsequent trials that haven't demonstrated that. CD19 is the kind of poster boy. It's where the CAR-T therapies have demonstrated sufficient efficacy that we now have commercial products that have been approved, licensed, and NICE approved, now rolled out in the UK, including in Manchester, both in the adult and the paediatric setting. You can identify antigens that are only accessible on cancer cells, so they're either in immune protected sites or only expressed during embryogenesis, and this includes the cancer testis antigens, and I'll touch on those in a moment. And then there's a whole array of other potential antigens, including tumor stroma components. These aren't tumor specific, but when you start to think about combining dual CAR-Ts that maybe can identify an overexpressed antigen alongside a tumor stromal component, will only be activated if both of those antigens are expressed within a similar location, then you can start to see how the kind of complexity of engineering may enhance efficacy. And we touched on neoantigens already. There's great interest in some of those high neoantigen expressing tumours around how we might be able to target those neoantigens with very specific products, particularly neoantigen selected TIL products. I'll touch a little bit more on CD19. CD19 is a great tumour antigen for CAR-T because it is expressed on many B-cell malignancies, and it is not expressed on either stem cells or elsewhere apart from normal B-cells. But actually we know from using rituximab over many years, patients are actually all right if they have B-cell aplasia, at least for a period of time. And there's strategies like giving immunoglobulins if required, if patients develop infections or problems with ongoing infections. So I'm sure many of you will be aware of some of the data that came out a few years ago now from CD19 targeted CAR-T cells, several high impact papers. This is just a selection of the trials that were carried out in B-cell acute lymphoblastic leukemia, mixture of adults and children. And these were generally patients who had exhausted all other treatment options and had very poor prognosis. And from those CAR-T papers, you can see that there's very high complete remission rates. And this slide's actually from CD19 targeting lymphoma patients in adults, the ZUMA-1 trial. But you can see not only were there response rates, but when you look at progression-free survival, there appears to be a plateau somewhere around the 40% mark, with durable remissions in some patients. And that really essentially curing patients who have no other treatment options is the sort of data that's led to registration and rollout of this type of therapy. However, there's loads of challenges. So you can get a feel these are complex, very costly, six-figure sums. We don't know how much it costs in the NHS because it's all subject to nice confidentiality, but they're expensive to deliver. And there's enormous practical challenges. Patients need to be inpatients for significant lengths of time in units that are JC accredited, etc., etc. And toxicity is not insignificant. The conditioning chemotherapy causes certainly neutropenia, sometimes anemia, thrombocytopenia. This graph just shows I think the top one's the absolute lymphocyte count, the white cell count, the neutrophil count, and the lymphocytes, and you can see that patients are profoundly neutropenic for a period of time. Cytokine release syndrome. I think everyone who's delivering CAR-T therapy now is becoming more adept at managing this. There's standardized protocols. We have tocilizumab. It tends to be used early. But cytokine release syndrome can happen over the first 2 weeks usually, and it's essentially a cytokine storm caused by release of inflammatory interleukin cytokines, and often can cause issues with hypotension, flu-like symptoms when it's mild, but in more severe cases, a kind of ARDS type picture. And then neurological toxicity appears to be quite common, particularly with CD19. This can vary again from just a slight word-finding difficulty up to encephalopathy and seizures. So it needs to be taken seriously and monitored very carefully. But once again, there's standardized protocols, and our ability to monitor and then manage patients early has been improving. So the success of CAR-T has largely been in the hematological and lymphoma setting for both adults and paediatric patients. And I think when we look towards solid tumours, we see significantly more challenges, not least because of the enormously immune suppressive tumour microenvironment that the T cells not only need to overcome, they need to track to the right place and then be activated, persist, and engraft. I'll not talk for long about this. It was one of the trials that we did. It was actually with the first generation CAR in CEA positive tumours, so in the adult population. And in this trial, we saw cell persistence and some enrichment for cells within the tumour microenvironment as opposed to in the peripheral blood. But we also saw significant toxicity. So we saw lung toxicity. You can see in this patient who subsequently recovered, there's patchy infiltrates. And whether this was just a cytokine release type syndrome that we've seen subsequently, or we did have some evidence of some upregulation of CEA, so cross-reactivity in the so on target but off tumour cross-reactivity. And so I think the landscape in solid tumours has been much more challenging. But what we've seen with the success of CAR-T in CD19 is a huge amount of work looking at new generations and new innovation, new engineering approaches. And there isn't sufficient time really to go through all of these today, but suffice to say that we're seeing a more sophisticated engineering, such as dual CARs that I mentioned earlier, that might target more than one antigen, making them much more specific to the tumor microenvironment. Combination with checkpoint inhibitors, where actually those checkpoint inhibitors are blocking the downregulation of the CAR-T as opposed to trying to generally stimulate the immune system, and approaches to actually engineering armoured CAR-Ts to increase the efficacy. I think one of the most exciting areas in paediatric setting, or applicable to the paediatric setting, has been the TCR T-cell therapies. And one example of this was originally developed by a company called Adaptimmune, but since bought by GSK, and we've now got the phase 2 trial open here at the Christie for the GSK trial. This is a patient with synovial sarcoma, so an adult patient in this case with multiple, it's a PET scan showing the black dots of multiple affected lymph nodes. And essentially, they had a complete remission to TCR T-cell directed T-cells directed against the testis tumor antigen NY-ESO-1. And this was awarded breakthrough designation in 2016 and has now gone on to, as I say, the phase 2 trial. We've just treated our first patient here at the Christie. Interestingly, I'm the CI for the UK for that trial, and the ethics committee allowed us to proceed with the trial in adults, but felt that it was ethically not right for children to be consenting to this type of treatment, because they felt they couldn't make the judgment about a treatment that could have the potential to be so toxic. I know that other investigators, including Sandra Strauss at UCL, who has this trial open, disagrees quite strongly with that, and we are going to challenge the ethics committee in relation to that. New data that's emerging, so another tumor testis antigen, so TCR, gene-modified T cells. This is still an Adaptimmune product, starting to see really promising efficacy. It's taken from around a year ago, but since then the data has persisted. In out of 12 patients with synovial sarcoma who'd failed first-line treatment, 7 of those patients had a partial response, and you're starting to see durability of efficacy as well. And that's the key to these really toxic, challenging treatments. I think just to get a progression-free survival advantage of a few months isn't going to be approved by NICE. It might be effective, but it won't be cost-effective. But if you can see in young patients, particularly durable remissions, then it starts to become really exciting. I'm not going to dwell on this, but one of the areas in childhood malignancies that's been actively explored, or a lot of interest in, is the use of CAR-Ts with local application, local regional application. This study was an abstract at ASCO in 2019, which was CAR-Ts against mesothelin in mesothelioma patients, so in adult patients, but combined with anti-PD-1. And I won't talk through the whole slide, but suffice to say that patients who received CAR-T and checkpoint inhibitor, they saw an objective response rate of 63%. So it wasn't a randomized trial, it was small numbers, but that is way higher than you would expect to see in mesothelioma patients with standalone PD-1. In this case, the CAR-Ts were injected locally. They did have some cyclophosphamide conditioning chemotherapy, but it was lower than the systemic doses that we normally give for CAR-T therapy. So significantly better tolerated. And when we look at the type of antigens that are being explored, obviously lots of hematological interest. BCMA is close to being approved by the FDA, as far as I understand. But there's also great interest in some of the solid tumor antigens that are applicable to the pediatric setting, GD2, neuroblastoma, glioblastoma. And some of these trials are particularly looking at local application, local regional application of CAR-T. Final couple of slides. I don't know how we're doing for time. We're a little bit over, but just to say, in Manchester, we have formed a collaboration, IMATCH, which some of you may be aware of, that cuts across both Christie, Manchester University Foundation Trust and the university, and commercial partners. And the idea behind IMATCH is really to scale up activity in the clinical setting. And subsequently, what we want to be able to do is pull through the discovery science into that delivery end of things. This is just one slide showing for ovarian cancer. One of the things that we're doing in association with the Manchester Cancer Research Centre is taking tissue from patients who are potentially undergoing advanced therapies, tissue and plasma, and having a kind of platform for how we might analyse this according to the scientific question that's being asked. So sample is prepared and purified in Richard Edmondson's lab, and then it's sent for various different analysis in different labs or commercial partners, and then brought together to form a kind of conglomerate of data that we can then apply. So the questions like, can we understand better toxicity in patients who are having CAR-T therapy? Can we identify patients who might respond or have a higher chance of response? So that's the TropStar platform that's being established, and we've just recently started collecting samples for that. But you can see how this is applicable to ovarian cancer, but in the future we could think about some of the other applications. It could be a platform technology that we could use in Manchester for whatever type of tumour, malignant tissue and blood samples we might look at, including paediatric cancers. I'm going to stop there. So I think there's some really exciting advances, and we're developing the infrastructure in Manchester to start to pull through that discovery science. And hopefully the next few years we'll see that evolving. So sorry I've overrun slightly, Martin, but happy to take a quick question if you have any.
I think in the interest of time, Fiona, we better move on.
Yeah.
But I don't know if you're able to stay for the breakout session later. It would be great if you could.
Yeah, I'll try, yeah.
Great, thanks for a very informative and interesting talk. Karen obviously is a professor in proton therapy physics and has a very active group based at the Christie site. So Karen's going to talk to us about potential development areas in radiation biology, because obviously radiation is one of our Manchester strengths. Thank you, Karen. I can see this but can't hear you, Karen.
Are going to.
Oh, there we go.
You should be able to see me, and I will start sharing.
Yeah.
Yeah. So Martin, with apologies, I've slightly modified the title to say because I've done it with protons, and it's a huge pleasure to be here. And thanks very much for the invitation. So to go forward. So I hadn't realised Caroline was going to use jigsaws. I'm about to use jigsaws. So what I'm going to talk about is a little bit about proton therapy. I'm going to show you quite a lot about our proton therapy research room, because what I wanted to do was talk about how engineering and biology come together to enable us to do something really different. Biological optimized treatment, that should say treatment planning. I'll talk a bit about that in a minute. I'm going to talk to you about flash radiotherapy, how we translate this, and then drug immunoradiation combinations. Now, when I started to look at this talk, I realized that I was going to go horribly over time. So I am not going to talk about biologically optimized treatment planning, but this is some absolutely fascinating work that's been led by Mike Merchant, and goes from looking at the individual DNA damage right through to how we can look at treatment plans and can we predict how protons will deliver the dose to patients, and can we optimize that? But I'm not going to talk about that, but you can get Mike to come and talk to you at some other time. Now, we've heard a wonderful talk from Fiona, and I would have, in a few years, well, I'm hoping in a year's time, through a Friends of Rosie grant, Amy Chadwick, who is a research fellow in the group, who's just got a grant from Friends of Rosie, will be able to come and talk to you more about protons and drug immunoradiation combinations. So again, that's something for the future. And I'm just going to transform the pointer. So that comes up in the future. So what I'm going to do today is to just talk about what's here. So I've got some thank yous. I have an absolutely brilliant group, and we started with about 3 of us in 2015. We're now up to over 40. And I should point out Amy, who has the Friends of Rosie grant, who will be coming back from maternity leave very shortly, and Mike Merchant, who I mentioned on the biological augmented treatment planning, who's ably assisted by Ron McCabe, the director of Christie Medical Physics and Engineering, and Adam Aitkenhead. And also I should mention Neil Burnett, who is our clinical lead within the group. But one of the really important things is working very closely with the clinical team. And without this close relationship with the clinical team and working in the Proton Centre, we wouldn't be anywhere. So huge thank you to them as well. It does sound a bit like the Oscars. I will get on to the talk, but I've also got to thank the people who fund this research, because again, without them, none of this would be here. And we're particularly excited. Amy's project is on paediatric cancers, particularly sarcomas, which has been funded by the Friends of Rosie, and we're looking at drug proton combinations there. And I should also mention the CRUK RadNet Centre. So here's our Proton Centre, and this started to treat patients in December 2018 and has continued all the way through COVID to be treating patients. Now, one of the things I wanted to draw your attention to is this, and this is the nozzle which delivers the proton beam to the patient. And this nozzle can move all the way around the patient through 360 degrees, so it can deliver beams at different angles. Now, bits of this nozzle will be coming in later on. Here's another nozzle and how we deliver it to the treatment bed. If we look at the clinical centre at the Christie, we have a cyclotron that accelerates the protons to about 2/3 the speed of light. And I should say, I think about 40% of our caseload is paediatric patients. I think that it's higher at the moment because of our colleagues in London haven't opened yet. And this is delivered to 3 gantries, which are used to treat patients. But you may see there is a space here. And this is known as the 4th room, sometimes the 4th gantry room. And we are incredibly lucky that the Christie charity, I should mention Ron McCabe, who wrote the business case for this, gave us the funding because NHS England won't buy concrete. So the Christie charity bought all of this concrete to build the research room, and also funded some of the beam lines that I'm about to talk to you. And this is so that we can actually do proton research. Why do we want to do proton research? Well, if we want the centre to stay at the cutting edge, and at the moment we have the leading proton technology anywhere in the world, we want to be able to have research that feeds through into the clinical centre. And that's why working very closely with the clinical teams is absolutely paramount. So this is our research room. And at the moment, as you can see there, it's just empty space. I'm now about to fill it. So this is, the beam comes through here, and this is our first beam line. So this is a schematic, this is beam line A. Beam comes here. We've got some very large magnets here, and that's because protons repel each other, and in order to keep them in a straight line, we need large magnets to put them back together again. So they stay in a focus beam. And they come along here, and then they come into a scanning nozzle. Now, this scanning nozzle is identical to those you saw in the clinical rooms, apart from the fact it sits on the floor. And that's because we don't need to rotate it around our samples. And then at the end, we can put different things so that we can do a range of different experiments. This is our radiobiology end station that I'm going to show you in a minute. For high throughput radiobiology, it's got a water tank after it to catch the protons. But we can start, we've designed this so it's flexible. So this part here is fixed, and then we can wheel in other end stations for other experiments as the need arises. And I'll talk about this second beam line in a minute. So that's the diagram. This is what it actually looks like. Here's the magnets. Let me just get to the magnets for you. except I've lost my pointer. Here's the magnets here. And this is the scanning nozzle sat on its edge. And then we come to, this is a hypoxia cabinet with a robot in it, which I'm about to show you. And this is looking into the room. So as I said, we built a robotic automated hypoxia end station. And part of the reason for doing this was we didn't want to irradiate one sample, switch the beam off, go and get the sample, take another sample in, switch the beam on again. We thought we would automate this. And this was so we could perform high throughput, repeatable experiments and reduce the error in proton radiobiology. If you look at what's out there in the literature, there's quite a lot of uncertainties. We've also put in a Versa liquid handling station, which allows us to then use different endpoints and use the facilities over at the MCRC. And we're also now beginning to add in live imaging so we can look at DNA repair kinetics. We've had a very close relationship with Don Whitley Scientific, who are just the other side of the Pennines, who have been absolutely brilliant. They've never done anything like this before, but they've worked very much with us. So again, what does it actually look like? Well, here's a picture this is at Don Whitley's site. This is as we were training the robot. And I've got 2 videos of how this works. So if you just bear with me, I think it will start moving now. So this is a robotic arm putting a sample, taking a sample out of the Versa and putting it back in what we call a hotel. This will take T98, 96 well plates, T25 flasks, and then it will go and get another one. And I'm just about to show you another video, and this is now with the under hypoxia. And you can now see it's gone to get, it's a 96 well plate, and it rotates it to the beam, it's irradiated, and then it puts it into the Versa so we can fix at different endpoints, and then it goes off and gets another one in a minute. So this is the automated hypoxia cabinet that we're just about to start using. It's all been trained and everything. Just a quick show, we've done an awful lot of commissioning. You can imagine that doing this sort of thing is not simple, and you want to make sure you've got the right dose and you're hitting the right point. And this is really very much where you get engineering coming together with radiation biology. This is showing the spot scanning, the water tank that the beam goes into. And I think if I show you here, this is a beam actually scanning in the research room a few weeks ago. As we started to do some of the commissioning for the dosimetry. Slightly slowed down because of COVID. We didn't get into the research room in March. First radiation biology experiments, not in the hypoxia cabinet, but this was just to start to look at some irradiations. And this is Ellen's work where she's been using different stains and using the high throughput imaging. This is a bit of work that comes from Charlie, who's shown here. Ellen, Charlie, Mike Merchant, John Warmanhoven, who is looking at micronuclei. So the second stage of this development is Beamline 2, because obviously you want to be able to do things in vitro first of all, but there is a lot of work that you will need to do in vivo. We don't want to design anything that's me too, so we asked our colleagues what was the absolute best, what would make the best system anywhere in the world. And so what we're doing here is we're designing a scanning nozzle for small animals, so we can do a much smaller beam spot, and we can image the animal so that effectively we are making this as close as possible to what is done clinically in patients, so that we can do effectively small animal work that is as close as possible to patients. Nobody else anywhere in the world has anything like this, and it's down to the team, particularly Mike Merchant and Mike Taylor, who have been fundamental to doing this design. And the advantage of having physicists and engineers as part of the group is that we're able to do this sort of thing. And so we're very keen to have a translational pipeline, as I mentioned earlier, which goes from basic research all the way through to clinical trials. And the Christie Charity funded all of this. So paramount to all of the research that we're doing is patient benefit, because if we can't see a benefit to patients, why should we do the research? We want to be able to translate all the research we do into the clinical centre. So I thought I'd now just talk to you about a new development which the team is very involved in, called flash radiotherapy. Now this all came to the fore in 2014 by this paper from Favardon et al. Working in Lausanne. Actually, no, they weren't. They were working at the Institute Curie, and Lausanne is also doing some work in this area. And they investigated lung fibrosis in mice. They used ultra-high dose rates over 40 Gray per second and compared those to conventional dose rates. Now, I should say this isn't the first time that flash has been done, and our colleague Jolyon Hendry, who was working in Manchester in the 70s, saw something very similar. But at that time, people were worried about the tumour control, and if flash spared normal tissue, would it also spare the tumour? And it was one of those subjects that went away for a while. But as I say, 2014, it came back. Now, this work was done with electrons. And it's been the normal tissue sparing that flash appears to give has been observed in vitro, but it's only observed in vitro at very low oxygen concentrations, which means that our hypoxia cabinet is absolutely designed for doing the radiobiology experiments that the community really, really needs, because nobody else has got the equipment to do it. It's also been seen in different species: mouse, cat, mini pig, zebrafish, and in different organs. And you can see here normal tissue complication probability versus dose, and you can see the effects of flash in normal tissue as opposed to non-flash. And if you start to look at the effects on the tumour, as you can see, flash appears to have the same effect on retarding tumour growth as conventional. And here we are. So flash appears to spare normal tissue while maintaining the tumour control. So you can see why there's lots of excitement about it. A study has also been done on mini pigs, and these were some studies done on cats. It's a phase 1 dose escalation trial on squamous cell carcinoma on 6 cats. And you can see that flash appears to be having a positive effect on those cats. Earlier last year, a 75-year-old patient with cutaneous lymphoma was treated with electron flash, 15 Gray over a time of 90 milliseconds. And I'll show you the response. So they saw less reddening, and after 5 months, although there'd been a little bit of edema straight after the treatment, after 5 months, it was looking pretty good. And there was, as I said, there was a little bit of edema, but that was only grade 1. So this is electron flash, and this was some work done in 2019. Started to report last week. Is the first proton flash trial on bone mets that's taking place at Cincinnati Children's Hospital, 10 subjects with painful bone metastases on the extremities. And that trial has just opened now. One of the reasons why protons seem to be so beneficial for flash is that the equipment we have now is just about capable of delivering flash beams with very little modification. But this is where the nozzle comes in again. And you may see this is a nozzle again. Measuring the very high dose rates and very short, and measuring that dose over a very short time requires a new type of nozzle and a new type of ionization. From base in the timescale of this project. I should point out, Verin have put quite a bit more money in it than the project gave them, but it came through that. So we've got, it's being tested, and Verin are now marketing flash-ready nozzles as part of their clinical sales. So what are the potential benefits for flash? Well, it claims, and experiments beginning to show, that it allows normal tissue sparing, and it looks, studies suggest that we can give 20 to 50% higher doses for flash to achieve the same toxicity as conventional. There appears to be no compromise on tumour control, although studies are needed for much longer times. Treatments are in fractions of a second. It means that motion management is less of a problem. The other thing we're finding with flash is it's a combination of the dose and the dose rate. So hypofractionation is really required in order to get the flash effect. So you need fewer fractions, shorter treatments, but particularly fewer side effects. So in principle, you can treat more patients within the clinical workflow, which should have impacts for economy and society. So very quickly, how does it work? Well, the stages of irradiation of tissue are physical ionizations, chemical reactions, and the biological effects. Now, I'm meant to be talking about the biological effects, but flash is very much in the chemical stage and is critically dependent on oxygen. And this gives rise to the oxygen depletion hypothesis, which was first postulated by Jolyon here in Manchester back in the 70s. And this is why flash appears to work in normal tissue, and we get the normal tissue sparing, because if you look at oxygen tension versus relative radiosensitivity, and you get a similar curve if you look at the oxygen kill ratio, and you can see that in normal tissue, which is generally more oxic, by putting the dose of radiation in, you deplete the oxygen and take it down here. Whereas in the tumour, you get a much smaller effect during the flash irradiation. So what we've done is we've built a model, and this model has a capillary, and then it has nodes which effectively go into the tissue beyond that capillary. And we can look at the way the oxygen diffuses during the treatment with protons. And we could, one of the nice things of using this cellular automaton is we can look at regions, diffusions in a multi-parameter space. So hopefully this will now work. So here's our the oxygen supply. The oxygen goes in, it's about to be irradiated, and it gets depleted. Now, the nice thing is, Ran looked at this with Matt Lowe, who's one of the other clinical scientists at the Christie, and said, oh, could we build that on proton spot scanning? And I'll show you that in a minute, but we now know that flash operates in a multi-parameter space. So it looks at the delivery parameters. So I've already said that dose as well as dose rate are important, but also the beam energy, the beam current. We know that the way the pulse is delivered and the pulse repetition frequency are very important, and that's why some people are finding it difficult to get flash to work. We know the radiochemical parameters are important, such as the depletion rate, the way in which oxygen diffuses, and our model uses effective diffusivity, so it matches what goes on in tissue, the oxygen tension in the capillary, and very important, the metabolic oxygen consumption rate. So if we take all of those, we can build them into a model of what actually happens in a treatment plan. And this again is thanks to Beth Rothwell, who's doing her PhD, and Matt Lowe. So what they've done is they've built Beth's model into proton therapy spot scanning. So we can superimpose the way the spots in a proton therapy treatment plan are delivered and build Beth's model. So we can look at different parts of the tumour and normal tissue and see what is happening with the spot scanning and the oxygen depletion, and then bring beams in at different angles and look at what's going on there. And we can look in at what's happening in normal tissue and compare that using the model to what's happening to the oxygen depletion. So this is right off the press. It's currently been submitted for publication. We're part of Varian's Fast Forward Consortium and are working closely with all these other Varian groups around the world. I should draw your attention, we're in the process of organising flash radiotherapy and particle therapy. There have been 2 flash workshops before, but this is the 3rd in the series that will be organised. We were planning for it to be in Manchester, but we're now going to plan to hold it in Vienna at Christmas. And we're hoping that it will be in person, but if it's not, it will be virtual. And we're holding it in collaboration with the people who've held the 1st and 2nd flash workshops, but also a EU project, UHD Pulse, and our project Inspire, which developed the flash capable nozzle. So coming to an end, I think I've shown you a little bit about proton therapy, quite a lot about the proton therapy research room and this partnership between engineering, biology, the clinical scientist, and that's how we can make a really excellent research facility. I've told you a bit about FLASH, and I hope I've shown you a bit about how we will translate that. So at that point, I'd like to thank you for listening. And Martin, I think you're over time, so if people want to ask me questions, I know I'm chairing one of the sessions.
Indeed. Thank you very much, Karen. You've brought us back up to time a little bit. So can I suggest we have a 10-minute break now, and after the break, Kate will open the chat, not the chat rooms, what are they called? The breakout rooms, and you can choose which breakout room you go into. So we have one on liquid biopsies chaired by Professor Brennan, another on radiobiology and radiotoxicity chaired by Karen, one on the immune environment chaired by Professor Rob Wynne, and then one on transcription factors chaired by me. And the idea there is to really go and think about what projects could Manchester lead on. So these are all areas where Manchester has a real pedigree in adult cancers largely. And it's really, this is now the time to think, can we have a leadership role in paediatric cancers? If so, how do we get there? What expertise do we have now? What expertise do we need? What relationships do we need to foster with other centres? And how would the path look to get from where we are now to where we want to be? So please, if you head off now, and we'll reconvene soon after 3:20, and then we can carry those on until, let's say, 4:05, and then another 10-minute break at that point. So when you come back, please choose your chat room, not chat room, breakout room. And then 2 minutes before the breakout rooms are closed, Kate will send a message, and then another message at 1 minute, and then on time the rooms will close. So if you're still talking at that point, you'll then come back and we'll have a discussion about where to move on from here. Okay, so we'll see you all soon. Thanks, everybody. We have another 10-minute break. On the agenda, actually, we've caught up time very well. I think we'll finish before 5. The workshop theoretically goes on till 5, but let's aim to finish a little bit early. So we have 10 minutes for the chairs to liaise with the people who are running the breakout rooms, and then let's reconvene at let's say 4:15 for a catch up. Okay.
Cool.
Maybe.
Cool.
Got some expertise there.
We're doing that outside of Manchester, so we could do that within Manchester.
Yeah.
Cool.
Right. Okay. Thanks.
But yeah, but please feel free to add to what I say.
Great. Thank you.
Everybody for sticking with us for so long. I hope you had as useful a conversation as we just had in ours. So this is just a wrap-up session really to go through the things we discussed in the breakout groups and think, where do we go next? So could I just ask each chair to go through and summarize the discussions in their group, particularly around the themes of what expertise do we have now? What are we missing? What are the projects we want to do, and where do we go from here? So the first one is the microenvironment. I can't remember who was chairing that. Who was chairing that? That was Rob.
How could you forget, Martin?
Sorry. I'm even looking at you.
Yeah, so I'm Rob Wynne. I'm a haematology consultant. I'm on the transplant program. So we had a constructive meeting, which I will summarize in a few minutes. So we talked about Manchester already as a centre of cellular immuno-oncology. So with several phase 1 CAR studies, commissioned CAR T-cell centre, allogeneic T-cell transplant centre for refractory leukemia and lymphoma. We talked that most of our collaborations, our add-on collaborations, are outside of Manchester. And so where we look to identify T-cell clones and emerging T-cell clones in graft versus leukemia, the collaborations are outside of Manchester. And we heard a lot of really interesting things from the group about brains and stuff. And so our working points were, first of all, pediatrics needs to get its act together and join Manchester Immuno-Oncology Network, which Fiona chairs and which I've been invited to, but we often don't get to. So we thought about maybe having a specific pediatric session where we talk about what immune therapies we use for children with refractory leukemia and perhaps other types of tumor and what our ambitions are. We also talked about a forthcoming clinical trial where we're looking at an immune-primed cord blood transplant protocol in refractory leukemias. And whilst we have money for add-on to see what the T cells that emerge are directed at and how they generate and maintain leukemic remission, we thought that maybe if we talked about that at one of the immuno-oncology networks, we might actually be able to do some of the work in Manchester that we're currently planning to do in collaborative groups outside of Manchester. And we talked about that Manchester and its cell therapies should join the more citywide biobanking around T-cell therapies, because although we do as much T-cell therapy as any other clinical institute in Manchester, then I don't think participate in those. So I think so those would be the beginnings. I think in summary, we are a large clinical centre. We are a large immuno-oncology clinical centre. Manchester obviously has all the laboratory stuff that we currently reach out beyond Manchester to do. So I think joining those two up is something that will come out of this workshop. And we've got a way that we might go forward.
You muted, Martin.
Sorry, Rob. And thanks, Rob. And is there somebody to lead on those actions?
So Fiona leads the IO network. I obviously lead the Cell Therapy Centre. I'm the CI on the forthcoming clinical trial. So I think joining that up will be good. But there are other people that need to be in this space that aren't represented at this meeting. So our tissue typing laboratory with whom we've done some of the T-cell work and the TCR work and the tissue typing and stuff. So they need to come to that meeting. So these are experts in clinical T-cell, allogeneic T-cell work. So if Fiona and I can, I think, generate a program that is of interest. We've talked a little bit through iMatch on this, Fiona. We did do a presentation, but it's mainly non-lab staff that come and things. So yeah, so I can bring our team, our large multidisciplinary team beyond the clinical team. We will, the lab teams, the research lab teams, including there's some xenograft work that we do currently outside Manchester with the T cells that we see that can clearly be done in Manchester. But I think it's about, in summary, it's about recognizing each other's expertise. We have expertise, but we don't use perhaps the expertise of the MCRC as we perhaps should have done. So I think there is a way forward, and there is a clinical trial onto which we can immediately graft some science that we can do in Manchester rather than outside.
That's great. That sounds really exciting.
Martin, sorry to interrupt. I don't seem to have a blue hand to raise, but just to say the IO network email address, I put it on the chat. So if anyone's interested to go onto the mailing list so that they can see when the meetings are happening, that sort of thing, it's there. And I'll take it back about the meeting to the committee that Rob mentioned.
Great. I think it's a shame JP isn't here. He was hoping to co-chair with me, but he's just had a baby, so he's on paternity leave. But I think he would have been particularly interested in the site-off work that we talked about as well. And I know he previously had a TIL study funded. So I think this is a very exciting space. I just wanted to get that word space in there somewhere. Great. Bernadette, are you able to summarize the discussions in the liquid biopsy session? Oh, you're on mute.
Unfortunately, we weren't a big group, so we had a nice discussion about the project that Alex presented, because I was the only person John Hickman attended, but we did talk generally. So we had a good discussion about that project and considerations around specific biology of childhood tumours and the fact that they tend to be genetically quiet, and therefore how we manage that. And we talked a bit about the technology. I thought of people that Alex could link up with that I have met that are involved in other aspects of it. And I think what we talked about is that the group for liquid biopsies are experts in liquid biopsies because of the newness to childhood cancer in terms of now and in this specific project. And of course, Ewing's is an all-age tumour. I think we need to collaborate a bit more with other biologists who are working specifically in other childhood tumours. And we talked about developing the methylation aspect of it, because that might be more important thing to look at in childhood tumours rather than, you know, most tumours don't have this nice fusion or a specific genetic marker to look for. So we talked about where this kind of work would go, and that while the expertise for liquid biopsies, and it's just by coincidence people like yourself and I are involved in clinical trials internationally in Ewing's, but that actually we should use the expertise, the linking in of the clinicians, but really we need to go beyond Manchester. So it's probably not quite what you wanted me to think about, but go beyond Manchester in terms of accessing then samples prospectively in clinical trials, because in order to follow on the nice picture that Caroline presented of the initial work, and then it becomes an NHS, if you like, test to the bedside, a lot more work needs to be done, and needs to be done in large prospective studies that we can access through Manchester CIs we can access into. And we talked about a bit more about the technology around. So there's lots of work doing on childhood panels for NGS in various studies. I talked a little bit about the link with SMPs and things like that, and how that interacts with these commercial platforms of Thermo Fisher and things, and what's the similarities and what the differences. So we talked about some work that could be done with regard to that. And what we felt really was that this project is a feasibility project to demonstrate that this is applicable in a tumour that they're not normally dealing with, and compared to sort of mainly adult epithelial tumours, and that doing some more work around linking with biologists who are interested in. And so the actions, if you like, is really around that core group that's been established to do this work, and how linking, using members of the core group, particularly people like yourself and myself, linked with biologists and others. So another tumour I thought of was, obviously, we talked about alveolar rhabdomyosarcoma, that's got a translocation, and there is biologists trying to do some work on liquid biopsies, and they could perhaps better link in with the expertise in Manchester. So that's where we got, because there wasn't really anybody else there but us.
That's a shame. I mean, again, it's a shame, you know, trying to coordinate everybody's diary is impossible. Unfortunately, Guy, who I'm sure would have contributed to that, is on for the week, so he was covering the wards. And actually, we had an email from Fiona Blackhall, who's the genomics lead for GM, whatever it is. She's the cancer lead for the Genomics Laboratory Hub, and she pointed out that all children will be getting whole genome sequencing from quite soon, which is another potential source of circulating mutations. So I think Fiona has plans to link in with the Biomarker Centre as well. about that, and that's another potential avenue for getting hold of samples and looking for low volume circulating mutations.
Mm-hmm.
Great. The next one is radiobiology. Karen, if you could summarize.
I think Karen's mute like me.
Unmute, please.
I've unmuted. Can you see my slide? Because we've got a slide.
Yep.
And in a minute, I am hoping to show that to you. Yeah. We had some lovely ideas, and if I've forgotten anyone's name, I apologise for that, because it's because I did it quickly. So Marianne came up with a lovely idea, which Anthony also sort of talked about, was the effects on teeth and linking in with dentistry of protons and photons, and that's not just for CNS tumours, that's independent of site. Obviously, Manchester has a huge amount of imaging data, and it was mentioned that we've got a new director of imaging who's been announced today, who I think is Stavru. We've also got some very good collection of outcomes. So, you know, putting all of that together, Neil Burnett talked to us a little bit about the RAPA project that he and Catherine have been very involved in, and is one of the, I think is the largest project in the world. But there was talk about whether we could go back to link stored blood and tissue samples for paediatric patients, simply to get enough data, and also linking to centres over abroad. We talked quite a lot about wearables. Mike Merchant in my group is already doing quite a lot in that area, and is particularly interested in impact on of radiotherapy on fatigue and sleep. But there was a new idea came up on a design of wearables for children. We then had variable wearables, or wearables, which I think was Anthony's idea. That's something that we thought would be a very interesting and novel project, both in the design of them, but also, you know, getting that out there. There was also talk about to take advantage of the structured way that the proton therapy service collects blood samples, and also the structured way to assess delineation. And we've got a lot of data there, and that was something, particularly for paediatrics, that we could interrogate and get more information out. There was the idea of a common toxicity to link with scan changes. I know Catherine West has been doing quite a lot of work with audiology, but doing more in that area, but also linking in with neurocognitive effects, particularly in paediatrics. And I know I've got Jill, Simona and Neil, but I know Marianne is also very working in that area. And then outcomes, particularly for protons, linking in with LET, RBE, but also proteomics and imaging. And finally, and we link into the liquid, that's meant to say biomarkers. Sorry, it says biopsies. So for example, if all of the liquid biomarkers start to give us early detection, then all our cancer incidence projection goes slightly awry. So actually we're developing new models to work with those liquid biomarkers, so that we can actually look at new models for changing cancer incidence projection. And that's something that Norman brought up. So that was us. Thank you.
Thank you very much. I mean, again, that sounds very exciting. There's a lot of potential work to do there. And people to do it. So in our group, we discussed the transcription factor. Again, it was quite a small group. It was Sam Butterworth, Kay Williams, Stefan Meyer and me, and Claire Dempsey was very kindly taking notes and helping make sure it ran smoothly. So we discussed, again, Ewing sarcoma. So Ewing sarcoma is driven by a fusion protein, EWSR1 Fly1. So we spent some time discussing what's known about that. In terms of what we have that's unique to Manchester, so Bernadette and I are respectively leading the main European first line and relapse trials. So we have access to the international trial community, access to patients and access to samples. And through that, there's also a very well-established biology network in Ewing sarcoma. We have animal models in Manchester. So Katie Finnegan has been working on an orthotopic osteosarcoma model, but is soon to start working on a Ewing's model. And Sam has the DNA encoded libraries that he talked about, which are, once they come in a couple of years, a very efficient way of screening large libraries. What we don't currently have is biological knowledge of Ewing's sarcoma in terms of having a good reporter assay for EWSR1, FLY1 activation, or anybody who's interested in the structural, the protein structure. So they're things that we would need to collaborate on. We already have some links to European and American labs who are interested in some of these areas, and there has been some work published on the protein structure of EWSR fly. In terms of funders, so we thought probably the main avenues would be, again, Friends of Rosie and other small charities, and then Cancer Research UK probably. If it wasn't cancer, then Wellcome Trust might have been good, but they don't so much support cancer research now. So again, it was a really useful discussion around one specific fusion protein. I mean, again, if we think of the transcription factors that drive a lot of childhood cancers, I think that the big pharma companies are going to be interested in things like MYC and MYC-N, and if there is an answer there, they will find it, not us. Whereas they're unlikely to be interested in the niche market of things like Ewing sarcoma. And it's a useful thing to choose because there are already academics in Manchester who are interested in it, and whatever we find there would likely be of relevance to other pediatric specific transcription factors. So it was a great conversation, and I will definitely take that forward. So we are coming towards the end. It's 25 to 5. I'd like to just open it up generally for discussion if there are other things that people want to say, because I don't want this to be, oh, Karen says, welcome to Fund Cancer, marvellous. That's good to know. I don't want this to be a monologue by me. It's a shame that some people were missing, particularly Guy Macon and JP Kildea, for the reasons I've said, both of whom were very interested in taking part in this and in the sort of things we've talked about. Before we finished, I just really want to thank Kate Vaughan, who's quietly sat in the background and has almost single-handedly organised this entire thing. I'd also like to thank the speakers and the chairs of the sessions, and Claire Dempsey, Kath Trigwell, Becky Holly and Nicole Davies, who sat in the breakout rooms and made sure they all worked, because I woke up lots of times last night having dreams about breakout rooms not working. So thank you to all of those people. Can I just open the floor for anybody who would like to say something? Please put your hand up or type or speak. Silence.
Martin, can I say something? I don't really deserve to say anything because I've only just come in for the roundup. But as a result of your workshops, and I listened to all the summaries, are you going to put together an action plan? What are the next steps?
So we've recorded this, the whole the meeting and also the breakout groups. Kate and I will do a summary document, which sort of very briefly summarises what was discussed and exactly what the next steps are and who we think needs to lead on those. I mean, we picked, there was a steering group that organised this meeting. There were lots of subjects we could have picked, but what we wanted to do was bring to the fore some areas where Manchester leads internationally on various aspects of cancer that don't necessarily have any paediatric focus in them, because paediatric cancer hasn't been part of previous BRCs or any of the really big infrastructural grants, and we want it to be part of future grants. But for that to happen, we need to have pilot projects that lead to bigger projects that lead to program grants that really increase our visibility. And this workshop, we hope, will be the thing that will kickstart all of that. So it doesn't matter if all of the areas we've discussed don't have progress, but we need progress in some of them.
Hi, Martin, it's Rob. And of course, the MCRC will help you package that. I think that, as I've said before, we're very interested in having elements of pediatric oncology in the new CRUK center renewal. it's certainly, you know, one of the reasons we wanted to co-sponsor this particular workshop and really continue to drive with you and look for the resources across the piece to continue to push depth into those USPs. So our strategic team is happy to help in that regard. As I said, even, you know, developing the 2021 and 2022 symposia and also lectures from international experts in this area to make sure that our brand is getting out there. And then also we'll do a collateral piece with you as well, which will be a PDF document that really speaks to our strengths in a very professional manner.
Great, thank you. Caroline, you were grinning then. I don't know if that was about something else or about
that was about something entirely different.
Oh, yes, okay. Don't know if that's
I'm on my 10th Zoom today. You're going to have to bear with me. I'm going a bit crazy.
Okay, so if there's nothing else to say, I think we can probably close it there, 20 minutes early. Thanks everybody who's contributed to the talks or the breakout sessions. I think it's been a really good day, and as I said, hopefully something that will kickstart the future. So you have 20 minutes free to do whatever you want now. Thanks, everybody.